Okay. So I just wanted to announce that there's an assignment, a homework assignment posted in Editat. It'll leave up at 1 today. So please make sure to do that. It's for points, but it's all, open book. Work through that. Keep on the PowerPoint slides that we're working through right now. Okay. And you don't have to show up to lab today, right? You have a video to watch. Really good one. So make sure to watch it. Well, potatoes. Good thing. So it's from the book. The book was written first by Michael Pollan, The Botany of Desire. I remember I first spotted it when I was in the airport and had a layover, and I was in one of the bookstores, and it's got a kind of a catchy title, The Botany of Desire. So it immediately caught my attention, and I had to buy it when I saw one of the chapters was about potatoes, because I was currently studying potatoes for postdoc. So really good documentary. Okay, so make sure to do that before next Thursday. And let's see, what else? So today I want to continue on with the moss information and bryophyte ecology. I will also get into hornworts and liverworts, and time permitting, I might introduce you to the next group of plants, which are the seedless vascular plants, because there's a lot to say about those as well. So here's the life cycle. Okay, so when you're studying this, I really encourage you to draw it out and talk it out as you're studying this, so that you get really comfortable with it, because this isn't going away. All right, and so here you see the two different phases of the life cycle, the gametophyte and sporophyte. And when you have to switch in between them, if you want to go from the gametophyte phase to the sporophyte phase, you have to have fertilization. Egg and sperm must fuse. And then once that happens, you're into the sporophyte phase. And then the sporophyte has to grow and develop and differentiate into a mature sporophyte and then start producing spores. But those spores have to go through meiosis before they're going to get released, before they're ready to go. And then once that happens, you're back into the gametophyte. So the gametophyte phase has half the number of chromosomes, haploid, or 1N, or you just put the letter N. And then 2N is referring to the diploid state, the sporophyte phase. So most of our bodies are diploid. You know, our body, as we walk around and live our life, most of our cells are diploid. It's only our eggs and sperm that are haploid. Okay, so we worked through that. And then we started looking at how to put that into context with the mosses. And so we were learning how mosses are typically unisexual. That means you're going to have a male moss, and you're going to have a female moss. Hopefully they're somewhat close together so that you can easily get transfer of sperm from the male to the egg of the female. And so the sperm production is happening in the antheridium, which is being shown up here in the top right. So that's just sacs of tissue where you're going to see the sperm being produced. And then in the female, you're going to have archegonia and just one egg in each archegonia. One big plump fat egg ready to go. So free water is necessary. You got to get some kind of splash event to get the sperm over to the egg. And if that happens okay, then hopefully you'll get fertilization. And then as soon as that happens, we start referring to that cell as a zygote. Then the zygote starts going through cell division, just lots of mitosis, and becomes an embryo. The embryo develops into a sporophyte. And remember, the sporophyte in the bryophytes, the sporophyte stays attached to the gametophyte. It's dependent for sustenance and nutrition on the gametophyte. And so as it's developing and growing and maturing, eventually its job is going to be to make spores. And then the last thing that has to happen is that meiosis event, and then they'll get free, and hopefully they'll disperse out into fresh habitat and start to grow and develop into the gametophyte. Okay, so we worked through that and looked at a variety of images. Hopefully you saw more of that in lab. And Once the snow melts around here, hopefully you'll be able to start training your eye and looking for the mosses. Remember, they're very petite, they're small, they don't have vascular tissue, don't have true roots. But that can give them an advantage in some respect, because if they don't have true roots, then they can pretty much grow anywhere. If they don't have to rely on water in the soil system, they can grow pretty much anywhere. And so that does give them some sense of freedom in that regard. So let's look at a few ideas with regard to the ecology. So again, small, compact, petite. They don't have xylem and phloem, so they don't have true roots and don't have to suck up the water from the soil. And so if you start looking around, even now with snow cover, if you look on people's roofs, you can see moss growing on rooftops or on cement walls or brick walls or rocks. I mean, if you just start looking, they're everywhere. And so the only thing we haven't found so far is no ocean dwelling mosses. We haven't found that, or bryophytes in general, not just mosses. But there are some freshwater ones, which I'll mention. And if you go to extreme environments like the Arctic or the Antarctic area, mosses and liverworts and hornworts, you can find them. And they actually do tend to outnumber the true vascular plants. Again, because they can just absorb the sleet and snow through their whole body. They don't need thawed out water in the soil in order to grow. They can handle long stretches of dark. So in those areas, sometimes darkness is pretty persistent for several months. Of course, freezing conditions. So they can tolerate that quite well. They can go dormant and just ride it out. And they know that many of the bryophytes do have partnerships with mycorrhizal fungi, even though they don't have true roots. Like when we think about mycorrhizal fungi, we tend to focus very much on the idea that the fungus is hanging out with root tissue. But just keep in mind that bryophytes don't have true roots, but the fungi can still associate with their tissue. And so and that makes sense in the idea that mycorrhizal fungi, they think mycorrhizal fungi helped plants establish on land from the ocean. And so mycorrhizal fungi were right there with the bryophytes as the bryophytes evolved. Yeah, I mean, it's similar to the air plant idea where you can see air plants just growing anywhere, no roots for the soil. So very similar in that regard. Air plants are vascular though. They do have vascular tissue, whereas bryophytes don't. Bryophytes just kind of absorb everything through their whole body. And if you think about like a new area that might have gotten scoured by fire, it's pretty destitute. One of the first organisms to come in can be the bryophytes, that group of plants, because again, they don't need soil conditions to be perfect because they don't really rely on the soil system for their water or their nutrition. So they can come into rocky areas or areas that have been burned over and can start to colonize. So especially when you think about rock formations, you can see that they can colonize. Similar to the lichens, they're very sensitive to pollution though, because their whole body is absorbing whatever is in the rain or sleet or the dust coming down on them. And so they can be sensitive to air pollution, in particular, sulfur dioxide in the air can really slow their growth and cause some problems. And they can also accumulate heavy metals, similar to lichens. Lichens have that issue too. And so they have reported fairly high levels, unfortunately, of zinc and lead, those heavy metals. They're good at hanging out. Again, like we said, they can hang out in the Arctic, Antarctic. They can be in desert conditions too, and which is kind of weird because I told you the other day they like moist habitats. But there are a handful that are well adapted to more desert conditions. And their strategy is often they'll go dormant for long stretches during the high drought season. But then when the monsoons come through and the rain lets loose, they can revive, come out of dormancy, and start growing. So they have kind of a patchy growth, and it depends on the rain season very heavily. These two images, you're looking at Tortula ruralis moss, and on the left, that's when it's dormant. And if you saw that, you might go, oh, it's dead. But actually, if you then douse it with some water, it greens up like what you see in the right image. And so even though they look dead, they're just sleeping, so to speak. They're dormant, but they can be revived and they can get very, very active pretty quickly too. So if you stood there and watched, if you poured your water bottle onto that patch and just stood there for like a half hour, you would see it green up and get active. Peat moss, you probably have heard of peat moss, and that is found in boggy areas. And we have peat moss here and bogs around here as well. And one of its claims to fame is that it can absorb a tremendous amount up into its body. And so 20 to 25 times its weight in water it can absorb. And so that's why if you pick up a bag of potting soil from Menards or wherever, very often if you look on the ingredient list, peat moss or sphagnum moss will be listed in there. Because if you mix that into soil, then when you use that soil for your plants, when you water the soil, the water gets retained and the sphagnum in the soil will absorb that water and then slowly release it. And so it's a way to kind of keep that potting mix evenly moist for long stretches of time. And that can really help with your plants that are growing in that. So sphagnum is adapted to really low pH. So pH of 3.0, that's very acidic. And for a moss, it can grow quite quickly. Of all the moss species, it's the quickest. And they estimate about 1%, a little over 1% of the Earth's surface is covered by peat moss. They say it's a source of renewable energy, but there's some debate on that. There's some groups that are trying to raise awareness that we have to be more careful how we harvest peat. Peat is harvested and grown for harvest in many regions of the world and need to be a little bit more thoughtful on how we deal with growing that for harvest purposes and for sale. Soil conditioner, that's the idea I was just talking about with the idea that you mix it into potting soil and it helps retain moisture. It is an acidic plant itself. It grows in a boggy area, which is very acidic. And so it actually is an acidic plant, and which can be very useful. And so, for example, historically, they used to pack diapers with, like your do-it-yourself diaper material. They would pack in sphagnum peat moss. The Native Americans were known to do that because when the baby urinates or defecates, the sphagnum peat moss would absorb that really well. And then the idea that you can stuff furniture with it, because it's acidic, and also they've isolated some other chemicals from sphagnum that is antiseptic. It's antimicrobial. So the combination of it being acidic and being antiseptic, it's good for treating burns or cuts. So if you're out hiking around and you've cut yourself accidentally, sphagnum peat moss would be a good first thing to do if you don't have any kind of first aid kit with you. Find some sphagnum peat moss and put that onto your wound. They used to use that during World War I. That was a very standard procedure to treat soldiers that had skin wounds. Oil spills, they've employed sphagnum peat moss for oil spills to help absorb all that nasty oil and try to clean up that area. And then even just like shipping fish, shellfish across country, when they're shipping it off for sale, they often pack the boxes with sphagnum peat moss and before they tape it all up and send it off, because the sphagnum will help keep the fish or shellfish fresh and hopefully keep any microbes from overwhelming and spoiling the fish. So lots of uses for sphagnum peat moss for sure. We've found a lot of reasons for it historically as well as currently. Here's just an image of a peat bog where you can walk on carpets of sphagnum peat moss if you are adventurous enough. You have to be careful because sometimes you can fall through and end up in the watery portion of the bog, but they're great fun to tempt fate and walk out on. This poor fellow was buried in a peat bog. This is over 2,000 years old. 2,000 years old, and you can still see all the skin, even the wrinkles in the skin. So normally when you bury a body for 2,000 years, it's down to bones by, in short order. So this is, again, saying how well sphagnum peat moss can preserve, because it's got all that antimicrobial, antiseptic activity. It didn't allow the microbes to decompose the body. So yeah, they did some work on this body, found that this person was about 20 years old when they died. And they think the person was deliberately buried in the bog. It wasn't like I don't know if the person was murdered and buried, I don't know, but they did discover it in a bog, and that's not normal. Usually you don't find a lot of dead bodies in bogs. So here they are harvesting peat moss over in Ireland, and you can burn peat, dried peat. If you take the harvest the peat moss and then dry it out really well, it burns really well. And so that image on the right is the idea of burning peat for a heat source. And so that is still done and can be done. If you're over 21, you might have some experience with whiskey. And so that is a very long tradition. This bottle that I'm showing here is Laphroaig. I think that's how you pronounce it, Laphroaig whiskey from Scotland, right? And so here we're looking at the idea of when you make whiskey, you use barley grains. And so what they do is they get a peat fire going and essentially roast and dry the barley grains in the presence of that peat smoke. And has anyone tried really, I call it peaty whiskey? It has a very strong sense of it. Well, to me, it tastes like I just shoved a bunch of sphagnum peat moss and started chewing on it. I mean, that's really it. I don't like it. I know a lot of people really enjoy it. Both my sister and I have tried just for fun, different whiskeys at that bar on. It's that bar on Crow's Nest. Landmarkin, thank you. Yes. And so tried a few different whiskeys, and that was one of them. I don't know. It's an acquired taste, I guess, but it's a big deal. It's part of their tradition of making this whiskey. So if you're over 21, you might want to sample that at some point to experience peat moss in a different way. So, mosses can be found all over the place. Here you're looking at a rock, as well as the more prominent mosses. So some mosses are super petite and very close to the substrate like this, where you really have to look. And then, of course, there's bigger ones, a little bit bigger, and they come up off the substrate. You can even go into cave areas and find mosses. And so this is a luminous moss that kind of gives off a glow in the darkness. So lots of different species out there. And so again, if you are out hiking around, really try to pay attention, because they're everywhere. Even in town, if you're just walking along the sidewalks or looking up onto the building walls or the rooftops, you can find the bryophytes, especially the mosses in this area. Okay, so that's the end of that slide set. I wanted to switch over into the second bryophyte side set, which is looking at the liverworts and the hornworts. And did you look at liverworts in lab, I hope? The Marchantia. Hopefully you were able to use a dissecting lens and take a peek at that. So we often show Marchantia in lab because it often produces lots of interesting parts of its body for reproduction. So now we're looking at the middle picture here, the liverworts, and the hornworts are on the bottom right. So I wanted to talk a little bit about them. So going back to the same phylogenetic tree where we had the Chara, green algae ancestor, and then you see the liverworts coming off, and then the hornworts are next, and then finally the mosses. So, the bryophytes are a very ancient group of plants. They were the first to really make it on land and make that shift from the ocean onto land. And the mycorrhizal fungi were right there helping them in that process. So here are two major ways that you will see them growing. They can be either leafy, which is being shown on the left for the liverwort. So that is a leafy liverwort. On the right is what they call a thallose liverwort. All right, and so Marchantia is a thallose liverwort. Okay, leafy, they are very petite still, but if you look at them closely, sometimes they can get confused. It's hard to tell the difference sometimes between a leafy liverwort and a moss. So what the giveaway is, whenever I'm hiking around and I look at something and I'm like, well, is that a moss or is that a liverwort? Look for whether or not it's bilateral symmetry or radial symmetry, that giveaway. So most liverworts are actually leafy, like what you see on the left. Leafy liverworts dominate. So the ones we tend to show you in lab, the Marchantia, that's actually a minor group of liverworts. It's just that they're really interesting, so we tend to show them a lot. But there's actually more leafy liverworts out there. And they exhibit what we call bilateral. We're bilateral. There's only one way to cut yourself in half to get a mirror. And same with liverworts. Whereas mosses, they're radial symmetrical, which means you can cut them in half any which way and you'll get a mirror image no matter how you cut it. And so when you look at liverworts, they can handle really moist conditions where it might be a little too moist for mosses. Liverworts love it. And so they like really moist conditions. And so if you really want to experience liverworts, go to tropical jungles because that's where you're going to find all kinds of different species. And when you look at leafy liverworts, if you look really carefully along the length, you'll see that they're little tiny leaves, which I put in quotes because they don't have true leaves, but we call them leaves. They overlap slightly, and there's two rows. There's always two rows of leaves, and so if you see that and you see that they're bilateral, you know you're looking at a leafy liverwort. So they dominate. There's more species of them, but there are the thallose. About 20% of liverworts are thallose, like Marchantia, and they tend to be more ribbon-like or flat and kind of lobed, like Marchantia does kind of look like a piece of liver when you look at its body, so that's why they gave it the liverwort word. And the reason we tend to show that in lab is because it can go through asexual reproduction and form these really cute structures for asexual, but then they also have sexual structures that are unique as well. And so those asexual structures are called gemma cups. They're little tiny, they look like a teacup that gets formed on the surface of the Marchantia body, and then there's little pieces of tissue. And that's the way they clone themselves. It's all done asexual. And then the idea there is to have those little pieces of tissue in a cup-like shape on the surface of their body. Then when the raindrops drop into that, it's a perfect design to get a raindrop to splash in there and very effectively bounce those pieces of tissue out and hopefully get them dispersed some distance away. So it's a neat little strategy. Okay, so let's look at the life cycle. Here we're going to look at Marchantia. So again, they put the gametophyte phase on top. So all the kind of peach lines here on the surface, on the top of this image, that's all the gametophyte phase. And then you see the dashed line, and what's below that is the sporophyte phase. So remember, with bryophytes, it's the gametophyte phase that dominates. That's what persists, and that's what you see when you're out hiking around. So here again, you can see that there is the male and the female gametophytes. They're separate. And if they're going to go through sexual reproduction, they have these structures. So the antheridophore, I'm going to blow this up a little bit. So, whoops, I wasn't trying to write there. The antheridophore. So anytime you see Phore, that means to hold up or to bear. So the antheridophore is just this stalked structure that comes off the surface, and they're just trying to put the sperm up high. They're just trying to raise it up a little bit. So if you look into the very top of that antheridophore, that's where the antheridia are located. And then remember, that's just stacks of tissue where you're going to get lots of sperm going on being produced in each antheridium. And so you have that going on with the male, and then with the female, they have a similar structure, slightly different top area to it. But again, it's an archegoniaphore. It's the soft structure called the archegoniaphore that raises up. And then if you look at the very tip area, that's where the eggs will be inside the archegonia. One egg per archegonium. And so it's kind of amazing when you look at this because the archegonia are kind of tucked under. So they're on the underside. It's kind of amazing to me that you ever get sperm and egg to unite in this situation. It seems like quite the challenge. But if you have that free water, the raindrops splashing just right, you will get sperm getting transferred from the antheridia to the archegonia. And if that happens, you're going to get fertilization to occur. So they're trying to show that event right there. And then you get the fertilized egg, which we call the zygote. So then that zygote just stays in the archegonium. It just stays put, but it starts to go through mitosis. Mitosis starts growing and growing and growing until you can see a little bit of a sporophyte peeking out there in that region. Okay, and then you get the fully developed sporophyte. And it's still attached there. It hasn't, it's dependent on the gametophyte. And then inside the sporophyte region, you have the sporangium tissue there. It's not nearly as obvious. If you go out hiking around, you have a hard time finding this. If you're hunting for a sporophyte, they're kind of tucked away. And you can notice them, but you have to be really vigilant and have really good eyesight. But eventually you'll get the spores being produced, and they have to go through meiosis, and then they'll get released. And then that word elaters on there, elaters, that is just those like brown strings of tissue that you see inside the sporangium. Those are what they call elaters. They think they're important for the spores to get. The elaters show up in a couple groups for plants. So they think they have this twisting action. The strips of tissue kind of twist and help kind of throw the spores when the sporangium breaks open. So same basic idea as what we talked about with the mosses. It's just there's a couple extra structures here to think about when you look at the antheridophore, the archegoniaphore, but you still have the antheridium with sperm and archegonium with eggs. So liverworts, again, really petite. The one that you saw in lab, very petite, low to the substrate it's growing on. This one's leafy. You can see there's two rows. They're kind of, there's that central midrib, and then the leaves come off that. So there's one row and the other row on either side of that midrib. And then the thallus type, this is more like what Marchantia looks like. Here's Marchantia. You can see those gemma cups, really cute little teacup structures. And then if you were to look inside some of these, you can see tissue, little bits of tissue. That one's pretty good, especially this one. You can see little blobs of tissue. So those will break free and get splashed out. Gemma cups are often called splash cups. Under the microscope, that's what they look like. So the cup-like shape and then the pieces of tissue. And so they're loosely attached so that when the raindrop splashes in there, that's quite forceful and certainly enough to knock out the gemma out of there. And then here's some images looking at the archegoniaphore and antheridia. So on the left is the antheridiaphore coming up off the surface of the liverwort, and then the archegoniaphore is a little bit different in its architecture there. But it's on the underside of the archegoniaphore that you're going to get the that's not the greatest picture that you can't tell these are better. So these are antheridiophores, and so the sperm production is happening up in the very top. Kind of looks like an umbrella. So at the top of the umbrella, you have the antheridia, and they're just chock full. All that dark blue is going to turn into sperm, lots of sperm. And then you have the archegoniaphore. If you zoom in right here, can you see that little yellow piece there peeking out? That's the sporophyte. So again, you have to have really good eyesight. If you're hiking along, bring a hand lens with you if you're really hunting for that, because that's hard to spot. So they're tucked under there where the egg, was sitting, and it got fertilized, and then that sporophyte starts to grow, but it doesn't get super big or long. Here's another patch. All that white that you're looking at, peeking out, those white things, that's the sporophyte peeking out. So again, you need some good eyesight to pick up on that. Here's a nice image here too. Okay. So the egg just stays put. Whether you're looking at mosses or liverworts, hornworts, the egg stays put. And once it gets fertilized, then the sporophyte starts to grow from that. And so that's why you see it's still hanging out in the archegoniaphore area. Stays attached, and the sporophyte needs to be getting food from the gametophyte. Under the microscope, it would look like this. So this is a archegoniaphore, and these two eggs got fertilized, and now you're seeing the sporophyte. And so this bigger area, this bigger, darker area that you're looking at, that's the sporangium. That's the area where you're going to see spores getting produced. Okay. So you need to get those spores produced. You need them to go through meiosis, because you want that to then break out of there, and hopefully air currents will take it and spread it into a new area where it can start germinating and growing back into the gametophyte. A few freshwater liverworts. If you own fish tanks or have aquatic systems, you might have purchased Reishi to help oxygenate and just beautify your tank. It's a very pretty liverwort. Okay, and so here on the left, you can see all the oxygen levels clinging to the surface of Reishi. So it's good for oxygenating in the close-up on the right here. And so that is, they're mass produced for that purpose. You find them in pet stores, or you can buy them for your tank to help oxygenate your tanks. I just put this in here because it's showing an aquatic liverwort where it has these kind of buoyant, air-filled parts of its body to let it float better in the water system. Real pretty. Okay, so liverworts are an interesting group of the bryophytes. And then the hornworts, those are the smallest in terms of number of species. There's not that many that have been discovered. And as far as I know, there's just one that's been discovered here in the more temperate regions of the world, in this area. And that one's Anthoceras. So there's only about 100 known species that have been discovered. And they look, if you look at the gametophyte, it kind of looks like a thallus liverwort. It kind of looks like Marchantia. But the reason they call it a hornwort is that when it produces a sporophyte, it's super obvious, and it's just this big spiky horn rising up off the rest of the body, the gametophyte body. And so they call that the horn, and so they got the nickname hornworts. So if you look at the gametophyte, the part that's just growing low to the substrate, you really have to hunt for the antheridia and archegonia. They're not obvious at all. They just kind of are partially embedded in the the gametophyte. And then they also have these like depressions or pores in their gametophyte tissue that the nostoc cyanobacteria like to hang out in there. So it's kind of a partnership where the nostoc cyanobacteria hangs out in those little depressions. It's very watery, so the cyanobacteria gets nice access to water, gets some nutrients from the liver or the hornwort, and then the nostoc, its claim to fame is that it can fix nitrogen. And so for the hornwort, that's great because the cyanobacteria is taking nitrogen gas from the atmosphere and fixing it into ammonia, which the liver or the hornwort and the bacteria can use. When the sporophyte is formed, once that egg gets fertilized and you get the actual sporophyte growing, it's obvious. It'll be this big spiky structure coming up off the surface of the gametophyte. And it's long-lived, more so than what we see with the mosses or the liverworts. The horn, the sporophyte horn, sticks around, and it can actually grow all season long. It actually has meristematic tissue that can keep it growing and growing. It stays green too. That's the other thing. It's capable of doing its own photosynthesis, but it still hangs out with the gametophyte. And so it's persistent. It's long-lived in terms of when you, especially when you compare it to the mosses or the liverworts. And then what happens is that whole long big sporophyte horn will just split lengthwise and just kind of peel apart lengthwise, and then the spores will get dispersed out of there. And they have elaters too that help twist and kind of knock the spores out of the horn and get them spread around. So if the sporophyte is being formed and you're out hiking around, you would notice it. With the naked eye, it's easy to spot. I've never noticed one though. In all my years hiking in this area, I've never stumbled on a hornwort. So if you find one, take a picture, let me know. So that's kind of what it looks like, where you have most of the gametophyte body is very flat. And again, you can't really see the Archegonian antheridia very easily. But once that egg gets fertilized, you'll get that sporophyte that looks like a horn. And then they're trying to show how it's splitting in half, peeling apart lengthwise, longitudinally. But if you wanted to try to find the Archegonian antheridia, like these are really powerful microscope pictures, and that's what you would need. You know, just with the naked eye, it's really hard to find the antheridia and Archegonia. So we won't ask you about this visually. Like, we're not going to put these pictures up and ask you questions about them because it's really hard to tell the difference between what's an Archegonia and what's an antheridia. So you need a powerful microscope for that. Here you see a successful fertilization where that egg did get fertilized. And now the sporophyte is growing out right from that fertilized egg. And it didn't even, it's long. This is just a partial capture. And then this image is trying to show you the idea that there's, down at the base, there's meristematic tissue that just all season long, it keeps growing and dividing to keep that horn structure, sporophyte, persistent. They're trying to show that it's green, even has stomata, which is interesting. So that's what it would look like if you're out hiking around. You might see something like this if you stumble on a patch of hornworts. And then if you take that sporophyte, spiky sporophyte, and look at it under the microscope toward the end there, you would see spores like this. So those purplish things, those are the spores. And then if you look really carefully under the microscope, you would see the gametophyte has a bunch of pores or depressions, and that's where you see the cyanobacteria hanging out. And they can do, the cyanobacteria can do photosynthesis, but the big benefit is nitrogen fixation. So if you take some of that tissue and look at it under the microscope, you would see something like this. And so Anabina is a cyanobacteria. And when it grows, it kind of looks like the cells just kind of stick to each other. So it kind of looks like a pearl necklace. So that's what you're seeing here. If you can see this, it's kind of in a circle. And so that's it's sitting in this cavity or pore. So that's the cyanobacteria. And there's another one right over here. There's one here, and one here. There's some more over here. So they're just hanging out there in a nice little swimming pool, so to speak, and fixing nitrogen. Okay, so this last slide is just a one-stop shop for looking at the three different groups, the mosses, liverworts, and hornworts, and kind of doing a comparison amongst them and what are their key characteristics. So this is good for studying when thinking about, what makes a moss versus what makes a liverwort a liverwort. And as you can see here, mosses win. There's more species of mosses out there that we're aware of than there are the liverworts or hornworts. Oh, also, I don't think I said this on Tuesday. Please make sure to start paying attention now to the phylum names of the plants we're starting to work through. So there, the mosses, they belong to the phylum Bryophyta. Bryophyta. Okay, so I want you to be able to recognize that on the exam. So I might say, what group of plants belong to the phylum Bryophyta? Okay. And then the hornworts, you see their phylum name is Anthocerophyta or Anthocerophyta. And then the liverworts belong to the phylum that they call Hepatophyta. Hepato is referring to liver. So if you remember that, hepato is liver. And so please start doing flashcards or something to match the phylum name to the common. Okay, yes. I think we went through most of this already. There's some detail in here that really not like you see there, it talks about phenolic material in the epidermal cells. Like, no, you don't need to know. Yeah, there's phenolic material out there. Some they where they live is good. Look at that one. That okay. Other questions on the bryophytes? Yes. No, not as clear cut. I'm particularly thinking of the hornworts. Hornworts tend to have both on one. Liverworts, this is my hazy memory, but liverworts I think are more unisexual, like mosses. So, but I can't swear to that. Double check. Other questions on bryophytes? The greenhouse technicians have been working hard at this. Hopefully they wheeled in some of at least one tank of their mosses that they've been growing for the greenhouse. And I know they have plans for the next plant sale that they're going to be offering they usually offer one more plant sale toward the end of the semester. I know they are going to make like little mini terraria with mosses. And so those will go fast, I'm predicting. So when the plant sales, pay attention to the flyers. I'll try to announce it lecture too, because those usually get snapped up really quick. So we bought little tiny glass vials with cork stoppers, and it's gonna be cute. So they're very creative. I really appreciate all their hard work. So I think we're gonna have a really fun plant sale. Okay. Can you get into EduCat, please? I just have a couple questions to ask you. It's open book. It says, look in your notes as you answer these couple questions. But let's take a break and do that.